Hello and welcome to BioCode. In this video, we will see how we can how we can provide attributes to a particular variable or particular atomic vector. For example, we declared a vector named as die, and we would like to also provide attribute to it so that a particular user or during our analysis, if we have forgotten what this variable actually does, how we can understand it or how we can actually uh, correlate it to a particular concept, that what sort of data or what really actually exists within this variable. So how we can actually provide attribute? These are known as attributes. What I'm describing right now, that anything that actually associates or that actually briefly tells you what this data is or individual data is known as attribute. So attributes really don't change the variable at is. For example, if we provide attributes to this variable, it won't actually change anything or it won't affect anything of that actually exists within this variable. Rather, it will be actually connected to it so that it can briefly present to the user what it actually is. So the most basic attribute that is utilized in bioinformatics or R language is known as names. So in which we provide names to to that data that exists in a particular atomic vector. So all you have to do is call the names uh, variable names function, that is built-in function that actually accepts a vector as a parameter. And once you have provided the parameter to the names function, all you have to do is call the concatenate function, and in that concatenate function you have to provide the name of the or names of the individual values. For example, in our dice we have 1 2 3 4 5 6, and each individual value should have a different name because we are providing the names attribute. So all we have to really do is we need to provide the name. For example, 1, and likewise 2 3 4 5